Okay, cells, DNA, and genetics part 2. So this actually has a focus on genetics and includes information about Punnett squares and common test questions that you can see in genetics. Okay, so here are some important people in history that form the basis of genetics as we know it today. Of course, there are so many people that have added to this knowledge, but these are some of the people that classically are taught that made observations that led to the study of genetics. But Charles Darwin looked at the evolution, development, and changes in organisms and said that they occurred through a process called natural selection. And that means that those specific organisms that survived had some advantage over other ones that may have been killed. So then what happens is they survive and reproduce, and whatever advantage, and we know that now that's a trait, will be passed on to the offspring. So it is through reproduction that genes are passed on to offspring, and over many years, organisms that survive and reproduce then are able to pass on their genes. And so cells, it developed from a simple cell to complex organisms. So for example, if you have a bird and it has a certain color, and that color allows it to be camouflaged in its environment, then that bird has an advantage in that environment to survive. And so they will pass on that color to their offspring, so then you'll have more of that color in that organism. So many traits like that evolved. It could be something like the height of an organism. It could be stronger muscles. It could be the speed. So anything that confers an advantage to that organism to survive, those are the ones that would be able to reproduce and pass on their genes to the offspring. So this is a slow process. It takes many, many, many generations. And really it's billions of years that we went from a simple cell to complex human beings. Now, Gregor Mendel, he did experiments on peas. So he was the first one that thought that genes come in pairs and are inherited as distinct units, one from each parent. And he made this observation through looking at pea plants and their characteristics after they were bred. So it could be the color of the peas. Some peas had wrinkles, some peas were smooth, so different characteristics, and he was able to figure out that based on the parents that were crossed, they may have a certain trait. And then the other important people in history are Watson and Crick, which are two scientists who discovered the structure of DNA. So again, DNA contains genes. Each gene codes for some protein, and then that protein has a function within the body. So genetics, there are certain terminology in here that you should know the definition of. One is a gene, which is a unit of heredity passed from a parent to offspring. Another is phenotype, which is a set of observable characteristics resulting from the interaction of gene with environment. So a phenotype is something that you can see. So for example, somebody's eye color, their hair color, their height. Some of them, things like height, is a complex interaction of many genes. It's not one gene that would determine that. But say eye color might be a specific gene. Then you have genotype, which is actually the genetic makeup of a person. So every person is unique. Every person has their own unique set of DNA. Although humans may be 99% similar, whatever the small variation is what will, give us a different appearance or so we'll have different characteristics. And then you have an allele. Now this is an alternative form of a gene. So an example is eye color. So you have one allele that may code for brown eyes, one that codes for blue, one for green, and one for gray. So which eye color you have is determined by the genes you get from your mother and the father. So the mother may have a gene, for example, for brown eyes, and the father may have a gene for blue eyes. And this is when you have the terms dominant gene and recessive gene. So the dominant gene is the one that, if it's present, will be expressed. So for example, brown eye is dominant. And so even if you have another gene from the other parent that may be a different color, if the brown eye is dominant, that's the one that will be expressed. So a recessive gene is one. Obviously, it's opposite of dominant. It will only be expressed if it has two of the same gene. So then we'll look at this Punnett square next to understand exactly what that means. So when you have two identical alleles, then it's called homozygous. If you have two different alleles, it's called heterozygous. So usually in genetics, the dominant gene is represented with a capital letter. So in this case, you have a capital A, so that represents some gene and characteristic. And so capital A means they have two dominant genes. If somebody has a heterozygous alleles, then you have one that's dominant and one that's recessive. So that's represented by a capital A and a small a. So the small a is the recessive. So the recessive will not be expressed. The dominant gene, the capital A, whatever that stands for, is what's going to be expressed. So in order for a recessive gene to be expressed, you'd need two small a's. And for example, blue eyes is a recessive gene, then you need one from the father and one from the mother in order to have the blue eyes. Okay, so this is the Punnett square, and basically what you do is whatever the mother's genotype is and the father's genotype, you put their genotype on one side, so you can see the mother's genes. In the first square it has a big B and a small b, so she's heterozygous. And then the father similarly is also heterozygous, a big B and a small b. So then what you do is you have the four squares, and whatever allele touches that square is what you put in the box. So in the first square you have a big B and a big B. So because they're both dominant genes, if we're saying that this represents brown eyes, then the person's going to have brown eyes. Then the next square over, if you go across from the mother's side, there's a big B, and then the father's side, it's a small b. So this is a heterozygous genotype, and because you have a dominant allele, again, this is going to be brown eyes. And then you go to the bottom boxes, and again, you'll see there's another heterozygous with a big B, so that's brown eyes. And then the last square is going to be the small b from the mother and the small b from the father. So now that you have two small b's, they will be expressed, and that is blue eyes. So whenever you have two parents that are heterozygous, like in this square, this is always going to be the same result. You're always going to get 1/4 of the offspring, or 25%, will have the big B, and 25% will have the small b, two small b's, and then half of them, or 50%, will have the big B and little b. And the reason they draw this out is just to remind you of what are the different combinations of genes that the offspring can have. So if so when this person has a baby, the child can have any four of those genotypes. And so 75% of the time, because you only need one big B, they will have brown eyes. And then only 25% of the time, or 1/4, they'll have blue eyes. So it's random. Whatever child is born, they could have any of these four genotypes. So that's kind of just a random thing that happens. And then if you go to the other Punnett square, if you change the genotype so the mother has two small b's and the father is heterozygous with a big B and a small b, what happens is half of the time you have two small b's, which is blue eyes, and half the time you have this heterozygous genotype, which means they would have brown eyes. So half the offspring have brown eyes and half would have blue eyes, or at least that's the chance that they would have brown or blue eyes. And again, every time a child is born, they can get any of these particular genotypes from the parent, and you won't know which one they're going to get because this happens randomly. And one more square that's not on here is if one of the parents was homozygous for the dominant gene, in other words, they have two big B's, then all of the offspring, regardless of what the other parent has, it's going to have a big B. And in that case, 100% of the offspring would then have brown eyes. Now, if the father was, say, recessive, then what would happen? Then 100% of the offspring would be of the genotype big B, small b, so they would all be brown eyes but heterozygous. If the father was heterozygous, big B, small b, then half the offspring would have the dominant gene big B, and half of them would have the heterozygous genotype big B, small b. So you can see, depending on what the parents have, then that would of course result in different genotypes and phenotypes for their offspring. So these are frequently test questions, and you may have the parent's genotype, and then they'll ask you to predict what percent would have, say, a certain hair color or eye color. And so you just draw this out, and then you'd be able to calculate, because it's hard to memorize all that. If they're heterozygous, they would have 25% of them would be big B, and 25% are small b. So this is just an easy way to draw everything out, and then you can see exactly what genotypes you could end up with in the offspring. Okay, so this is called a karyotype, and what this does is the chromosomes here you can see are labeled from 1 to 22. So that's how many sets of chromosomes they have. So chromosome 1, for example, there should be 2 of each of the chromosomes, because one is from the mother and one is from the father. And then you have the sex chromosomes, an X and a Y. So if a person has an X and a Y, that means they're male. Just carrying the Y chromosome makes somebody a male. If this person had 2 X chromosomes, then they would be female. So as we talked about in the previous lecture on cells and DNA, when you have the egg and the sperm come together, half of them carry the mother's genes and half carry the father's. So in this case, you'd have 23 from the mother, 23 from the father. And the 23rd one is the X and Y chromosome, or it could be an X and an X chromosome. So every time the cell divides, all of these are passed on to all the cells within that organism. So sometimes you can have errors that happen. An example is when you have a trisomy, which means you have 3 of a particular chromosome. So if you have 3 of chromosome 21, for example, this results in what I'm sure you've heard of as Down syndrome. Now, most of the time when you do have some kind of a trisomy, or say a cell that has only 1 of a particular chromosome, that can be lethal, and that would cause the cell to die. Now, if that happened in the initial first cell, or in any of the stem cells really, and then that further divide, that can result in a miscarriage or a loss of the fetus, in fact, before it is fully grown, because those would not be viable. So Down syndrome is one that the offspring can still survive with an odd number of chromosomes. Although, as you know, they do have health issues, things like decreased IQ and so on. And And the actual name for that is called non-disjunction, which means that two of the chromosomes are stuck together and they're actually unable to separate. So that's a problem with non-disjunction. Okay, this is something else you frequently encounter in genetics when you're taking a class at least, and these are called pedigrees. And what this represents is people in a family that have a specific disease. And I mean, it can also be used to talk about a characteristic or even in plants that you breed. But for example, here you'd have the generations 1, 2, and 3. And the squares always represent males and the circles always represent females. And if you have a line drawn through it, like in the second generation, this male has a line drawn through, that represents someone that has died. And then here in the third generation, you have a female with a line drawn through, and that's a woman that has died in this generation. And if they're shaded gray, that means that they have a particular trait or disease that you're looking at. And if they're unshaded and are white, then they're not affected. Now, the purpose of this is to kind of get an overview of what their particular family tree looks like, what disease, and how the disease is passed from generation to generation. And so in the bottom here, there's another pedigree, and it asks what pattern of inheritance does this trait follow? So frequently you may be given some information, you may even be given a genotype of somebody in the first generation, and then you have to figure out how this was transmitted. In the case of this second pedigree, there's a red circle, which is an affected female, and then she has 4 children, and none of those children are affected. And then in the third generation, a male is affected, and the person he marries, and that's what the bar represents, is the person that's been mated or married to, and then the line that goes down is, of course, her children. So that female is also not affected, but yet the one child is affected in the third generation. So most likely this represents a recessive gene. So if the grandmother here that's affected has the recessive genes and the father had a dominant gene, then the second generation would not be affected. So the male likely here is heterozygous, and then the female is likely heterozygous, meaning they have, you know, a big A and a small a. And then the third generation, this particular male received a small a and a small a from the parents. So then they are affected. So like I said, these are frequent test questions. You just kind of have to know what the symbols represent and how to draw them into generations, and then which two are mating or married. And then you just kind of analyze based on the mode of inheritance and try to figure out what may be going on. So up to now we've been talking about dominant and recessive genes, and it's actually more complex than that. There's genetics is actually really complicated, and there's a few other terms. For example, incomplete dominance and codominance. And incomplete dominance means that one gene is not, well, as the name implies, is not completely dominant. Some of the characteristics of the other gene or allele also shows through. And then in terms of, for the term codominance, it means that both alleles are expressed in some way. And we're not talking about colors specifically, but for an example, your blood group, some people have an AB blood group. And so if you have one allele that codes for the A blood type, and then the other parent has the B blood type, when you receive one of each allele, you'll be codominant and express the AB blood group. Now, within that ABO system, the O blood type is actually recessive, so if you have an O, it will not be expressed. And so if one parent has an A and one parent has an O, you would have AO, and the O would not be expressed, and you would show as a blood group A type. So, so there's many other complex interactions like that. And I just listed a few diseases based on their type. For example, recessive diseases include hemophilia and sickle cell. Sickle cell disease is autosomal recessive, and then hemophilia is actually an X-linked recessive gene. And then other X-linked recessive disorders are color blindness and Duchenne's muscular dystrophy. And then some diseases are based on mutations in the DNA, for example, cystic fibrosis, hemophilia, and sickle cell disease. So there's some change in the DNA that leads to a change in the N protein that results in these diseases. So that's why this series, if you go in order, it's good to understand what is DNA, how it codes for a protein, and how changes in the DNA can lead to an altered protein that can cause a disease. Now, in the case of sickle cell, it actually has, although it's a disease, it does have a survival advantage, which is why you still find it in populations, especially in Africa. The red blood cell sickles, or in other words, it changes shape and has kind of a sickle shape, and that the advantage of that is that they're less likely to get malaria. So this is actually a survival benefit of having this sickled red blood cell. So they are more resistant to getting malaria. Malaria is caused by a mosquito that infects the host with a parasite that then goes on to infect the red blood cells and can cause severe symptoms of fever and rupture of the red blood cells and hemorrhage and so on. Okay, well that concludes genetics. So we just talked a little bit about the history, kind of the modes of inheritance, the Punnett square, the pedigree. Those are all kind of common test questions. I mean, that's just the very basics. And this summary slide just goes over what we talked more about in the previous lecture about the chromosomes, how we get them from the mother, father, and then how that affects the cell type, and how genes are passed on to the daughter cells, etc. So you can just read over this on your own. And the next one probably will just be a little bit in more detail about proteins, because I couldn't really discuss enough detail about it in the previous lecture. So like, subscribe, and share.